welcome to today's Life Sciences lesson. My name is Dito Makwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So, in today's lesson, we are looking at our paper 2 revision. We are looking at part 10, and we are going to be looking at question 2 here. So, remember, we're breaking down our question paper into two parts of section B, right? We are looking at question 2 for 50 marks, and we are also looking at question 3 for another 50 marks. And just a reminder, we also have section A, which is 50 marks as well. In section A, remember, we have our short questions, we have our multiple choice, we have our biological terms, as well as the matching column and a few short answer questions. Right, so just a reminder of your topics for paper 2. You must make sure you remember these so that you prepare for the correct topics for the correct paper. Right, DNA, code of life, that is 27 marks. And then for 21 marks, we have meiosis. And then we have genetics and inheritance for 48 marks. And lastly, we have evolution for 54 marks. Right, okay, by the end of our lesson today, you should be able to describe how a change in the DNA leads to the formation of a different protein. So here we'll be looking at some mutations that may occur leading to changes in the DNA sequence, right? Now, when there is a change in the DNA sequence, there is definitely going to be a change in the mRNA that is formed during protein synthesis, right? Now, that mRNA that is different as well would lead to different tRNA anticodons that are going to be brought in together with the amino acid. So that amino acid is also going to be different, meaning that the amino acid sequence is also different, leading to a different protein that is going to be formed, especially if the triplets do not code for the same amino acid. Also, keep in mind that the triplets may code for the same amino acid. So even if there is a change or a mutation, it might not lead to any changes if the codons that have been changed or the triplets that have been changed are still going to code for the same amino acid. So we will have a look at that. Right. So you should also be able to tabulate the difference between DNA replication as well as transcription. So we know that DNA and transcription have some similarities. However, you should be able to know the differences, which means that if you look at the process of DNA replication and you look at the process of transcription, you should be able to know that these are two different topics, right? Or two different processes. And then also, if you read about DNA replication and you read about transcription, you should be able to see that these are two different processes. Okay, you should also be able to describe sex determination in fruit flies. You should also be able to complete a genetic cross on sex-linked characteristics. This is very important. We will look at it right at the end, whereby we are looking at the fact that when you do genetic crosses that are not sex-linked, autosomal disorders or autosomal characteristics, these are very different from sex-linked characteristics. That is very important. We are going to look at that just so you can have a reminder of that and be able to use the correct format there. And then you should also be able to describe how non-disjunction leads to Down syndrome. All right, let's have a look at our first question. Our first question is question 2.1. And it says a sequence of nitrogenous bases in a DNA molecule is shown below. We have CCC, GGT, and TCA. So this is a DNA molecule. One other thing that you can see here that indicates to you that this is a DNA molecule is that we have the thymine. Remember, in DNA, we have thymine, and in RNA, we have uracil. 2.1.1, write down the mRNA codon sequence that reads from left to right from the DNA sequence above. Now, this one is straightforward. You are just expected to give the mRNA codon sequence. Remember, when we have this is a triplet, right? Now they are saying you must give the mRNA codons. Now, here, these are codons. This is a DNA triplet. And then we also have the tRNA anticodons, which is not asked for in this question. So let's have a look at our answer. So we know that C pairs with G, so it means it will be G, 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 and then G pairs with C, so we'll have C, C. T pairs with A, so it will be C, C, A. And then T again pairs with A, C with G, A pairs with U. Why does A pair with U? We are looking at RNA. In RNA, we do not have thymine, but we have uracil. Therefore, it will be AGU. All right, so this would be our answer for 2.1.1. Okay, the GGG, CCA, and AGU. Okay, let's move on to 2.1.2. It says the table below shows the tRNA anticodons and their corresponding amino acids. Right, so in this table here, we have the anticodons and then we also have those amino acids. Right, now we are given a DNA sequence. You can easily pick that up because here we have thymine and in the tables we do not have any thymine. So that should show you that the amino acids are not coded for by the DNA sequence that we have been provided with, right? And that means that you should then use your DNA triplets and your mRNA codons to form that tRNA, right? So here we are going to have the tRNA anticodons. Remember, these are anticodons. We have triplets in DNA, codons in mRNA, and anticodons in tRNA. Now, we have formed the mRNA sequence using the DNA. So now from the mRNA, we can translate to the tRNA. GGG will pair with C, 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 and then CCA will be GGU, and then AGU will now be UCA. Right. Okay, so this is our tRNA. Let's just quickly take this and write it down on the other side. So we have CCC and GGU, right? So we have CCC, GGU, and lastly UCA, and then we have UCA. Okay, the question says write down the amino acids in the correct sequence that would be coded for by the DNA molecule above. So remember, the DNA molecule codes for the mRNA codes for the tRNA, which is what we have here. So now we are going to take this tRNA sequence because these are our anticodons and pair it with the relevant amino acids that the anticodons bring to the ribosome during during protein synthesis, during translation. So now these are our anticodons. We're going to look at them from the table and then pair them with the correct amino acid. CCC, we have glycine. GGU, do you have GGU? Yes, we have proline. And then lastly, UCA. Let's have a look at UCA. There we have it. It is serine. So that is how we would answer this question over here. So that will be your 3 marks. Okay, let's have a look at that. Glycine, proline, and serine. Okay, let's go back to 2.1.3. During transcription, the first triplet in the DNA sequence changed from CCC to ACC. So this means that our C has been replaced with A. So instead of having, okay, CCC, ACC, instead of having GGG, we now have UGG. Then the question says, explain how this would affect the protein that is formed. So our CCC has now changed to ACC, meaning that we are now going to have an mRNA of UGG instead of GG. Right, now if we take this to being a tRNA, we are going to have CCC, and we are going to have ACC. So this is what we're going to have instead. Now, CCC will then change to ACC instead. And what amino acid does it code for? ACC codes for the amino acid tryptophan. So this means that instead of the amino acid glycine, we are now going to have the tryptophan amino acid, and that will definitely lead to a different protein being formed. Right? So the codon would change to UGG, the anticodon with ACC will bring the tryptophan amino acid instead of the glycine amino acid, right? The sequence of the amino acids will change and a different protein will be formed. So it is very important that when you give this kind of an answer, you don't just give a generalized answer whereby you are going to say the mRNA sequence will change, therefore the tRNA anticodons will also change, and it will bring in a different amino acid, and the protein will be different. Be specific. Say that our codon has now changed to UGG and the anticodon has now changed to ACC, which then gives us a different amino acid, specifically tryptophan instead of glycine, which then means that we have a different protein that is going to be formed. Do not just generalize. Make sure that you are specific with your answer there. Okay, 2.1.4, it says describe the role of tRNA in translation. So remember, translation is the second part of protein synthesis that takes place in the cytoplasm. So now, during this translation, the tRNA is responsible for bringing in the relevant or the correct amino acid that is complementary to the anticodon on the tRNA, right? So each tRNA carries a specific amino acid to the codon on the mRNA. Remember, the anticodons pair with the codons on the mRNA. So these tRNA brings in the relevant or the specific amino acid onto the ribosome to pair with the codon on the mRNA. All right, 2.1.5 tabulates two differences between the process of DNA replication and transcription. As I've said, these two processes are similar. The description is the same. The only difference is that during DNA replication, both of our DNA sequences or both of our DNA molecule strands actually serve as templates for the formation of the new DNA molecule, right? However, during transcription, which is what we're differentiating, during transcription, only one DNA will serve as a template. Why is this? Because we only need one mRNA to be formed. We do not need two DNA mRNA molecules. So only one mRNA, therefore only one DNA will serve as a template. And we also have the fact that in DNA replication, we look at the DNA nucleotides that come in to match or to form complementary base pairs with the original DNA. However, during transcription, it is not a DNA nucleotides, but it is RNA nucleotides. Right, so the free nucleotides. And then during DNA replication, again, the whole DNA will unwind, whereas only a part of the DNA will unwind in transcription. And lastly, the A pairs with the T and A pairs with uracil in transcription. Make sure that you have a table. Write this in a table form. Therefore, you will be able to get that 1 mark for the table. All right, let's have a look at 2.2. We are looking at the diagram of a karyotype of a male fruit fly and the karyotype of a female fruit fly. It says here, the diagram shows the karyotypes of male and female fruit flies. So there we have our fruit fly, and we have the XX for the female, and we have the XY for the male, and then 2, 3, and 4 are the same. Okay. 2.2.1, it says state what is meant by the term karyotype. So remember, when we're looking at a karyotype, we are looking at the arrangement of chromosomes of an organism within the cell of an organism, right? So here we're looking at the arrangement, the appearance, as well as the number of chromosomes, right? So those are very important. The appearance and the number is very important, because remember, different organisms have different number of chromosomes, right? So the number and appearance or type of chromosomes in the cell of an organism, that is what a karyotype is. Okay, what is the diploid number of chromosomes of this species of fruit fly? Let's have a look. So here we have 1, 2, 3, 4, 5, 6, 7, 8. So the diploid number would then be 8, meaning that the haploid number would be 4, right? So 2n is 8 and the n is 4. 2.2.3, it says describe how sex determination in fruit flies is similar to humans. Now, if we have a look at these diagrams here, we would see that in males we have XY, which is what we would find in a human male, and in females we have XX, which is also what you would find in a human female. So that is the similarity here. Females are XX and males are XY. 2.3, it says the gene for eye color in fruit flies is carried on the X chromosome and therefore is sex-linked. The allele for red eye color is dominant over the allele for white eye color, which is what we have here. So this is recessive and this is dominant. So what do we have here? We have white, which is recessive, and we have red, hence we are using the letter R, which is dominant. Right. 2.3.1, it says state Mendel's law of dominance. So remember, when we look at Mendel's law of dominance, we look at the fact that when two organisms with contrasting characteristics, right, that are homozygous, so they're both homozygous and they are contrasting, when those two are crossed, the offspring are always going to take the characteristic of the dominant individual, right? Which then shows us that the dominant one is then going to overpower the recessive allele, right? So for example, if we are looking at XR and X small letter r, and they are both homozygous, it means that all the offspring that we are going to have are then going to be red, because X capital letter R will dominate over the X small letter r, right? So when two homozygous organisms with contrasting characteristics are crossed, all the individuals of the F1 generation, those are the offspring, will display the dominant trait, right? Or you can say that an individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype. So when you are heterozygous, because you have the X capital letter R and the X small letter r, the capital letter R will always dominate over the small letter r. Therefore, in your phenotype, you will show the dominant phenotype. So 2.3.2, it says a heterozygous female is mated with a white-eyed male. Right. Remember, as soon as they say heterozygous, it means that this individual is the dominant one or is carrying the dominant characteristic. Why? Because you cannot be heterozygous and have the recessive phenotype, basically, right? And then this one is white-eyed. We know that having the white eyes is the recessive trait, right? So now, because this is the female, it will be XR, X small letter r, right? Because that is the female, right? And then the male is white-eyed. For the male to be white-eyed, it means that the one X is going to carry the small r. And then remember, in males, because of the male's sex determination here, we know that it is XY, right? So we know that the Y chromosome does not carry any allele, so it is going to be X capital letter, small letter R, and a Y that does not have anything, right? Then it says use a genetic cross to show the possible genotypes and phenotypes of the offspring, right? Let's have a complete genetic cross here. Remember, guys, make sure that you write the full cross. So you start with your P1 and your F1 down there, right? And then with your P1, you have your phenotype, and then you have your genotype, and then you have meiosis, and then you have your gametes, and then you have fertilization, and then you have for your F1, let's just bring the F1 a bit up. You have your genotype again for the F1, and you have the phenotype for the F1. Right, phenotype, it is a red-eyed female. Red-eye female crossed with a white-eye male. Then the genotype. Here we have X capital R heterozygous, X small r crossed with a white-eyed male, X small letter R Y. Then the gametes. Remember, the gametes are the same as your genotype, just show that you have separated them. And then X small letter R Y. Fertilization happens, you're going to cross them. When you cross them, you start with the first one, X, and that one. Therefore, you have X capital R, X small r, and then X capital R and Y, and then X small r, X small r, and then lastly, X small r, Y. Right, so now let's have a look at the phenotype. We have a female here who is red-eyed, red-eye female. And then we have a red-eye male as well. And then we have a white-eye female and a white-eye male. So we have one of each: white-eye female, white-eye male, red-eye female, and red-eye male. So as you can see here, with the male, do not forget that when we're looking at sex linked, it is linked to the X chromosome. So we do not have the Y chromosome carrying any allele there, right? So there we have it. This is how it is going to look. You get a mark for your phenotype, you get a mark for your genotype. In this case, you are getting 2 marks for each genotype, meaning that even if you make a mistake here, you would still get a mark. For the genotype of the female, you get only 1 mark. For the gametes of both the male and the female. And then it says in the question previously, to give the possible genotypes and phenotypes. So that is what would be your compulsory answer there. So as you can see, it is starred, meaning that if you do not give this, you will not get the marks. So your genotype and then your phenotype. Remember, P1 and F1, they have a mark. Meiosis and fertilization also have a mark. You can also use a Punnett square. As we can see, there is the Punnett square. You follow the same process, and instead of showing the crosses, you would then show a Punnett square. Right, 2.4 says the histogram shows the range of heights in a sample of rose plants. Okay, so we have the heights here, the different heights, and we have the number of roses that have that height. For example, the height from 20 to 25 centimeters, we have 2 roses. And then 31 to 35 centimeters, we have 9 roses. And 36 to 40, we have about 4 roses. Right. Now let's move on to the questions. It says, what type of variation is shown by the height of roses? Because we can see there's a range from 20 all the way until 45. And we say there's a range, it means that this is continuous variation, because there is continuation in the phenotype that we're looking at or the characteristic that we are looking at. So variation here is continuous. Okay, give a reason for your answer in question 2.4.1. So as I said, there is a range of different heights from the lowest to the highest, right? There's a range of intermediate phenotypes or heights. Okay, 2.4.3. A rose flowers with long stems sell for the highest price because the long-stemmed flowers looked beautiful in a vase. Plant breeders select plants with the longest stems to interbreed. And these long-stemmed plants do not necessarily survive in the wild because wind can bend and break them. Okay. Explain how this practice of interbreeding long-stemmed flowers is an example of artificial selection and not natural selection. So remember, with natural selection, nature is going to select the organisms or the flowers that are good enough or favorable to survive within that environment, right? However, in this case, it is not nature that is selecting. However, it is the breeders or it is human beings who are going to select the organisms that are better suited for the interbreeding and those that contain the favorable characteristics. So plant breeders or people are the ones that select the characteristics and not nature selecting. So it is not nature that selects the organisms with the favorable characteristic. They select those characteristics that are desirable to them. So it is the human beings who select the characteristics that are desirable to them. That is why when we talk about artificial selection, we talk about how that is something that will now satisfy human needs, right? And is not beneficial to survival. So it is just only beneficial for the needs of the human beings. However, it is not beneficial for survival. Okay, let's look at 2.4.4. When rose plants with yellow flowers are crossed with rose plants with red flowers, the offspring all have orange flowers. So now we have yellow, red, and orange. Now this starts to tell us that we are no longer looking at complete dominance because there is nothing that is dominant over the other. Remember, when we're looking at complete dominance, red would probably be dominant over yellow and the offspring would be more red than yellow. Or however, in this case, because we are getting a new color, we are getting orange. It means that we are not looking at complete dominance. However, how do you know which one we are looking at? Are we looking at codominance or incomplete dominance? Right. We are looking at incomplete dominance. How do we say look at this? When we're looking at codominance, both the red and the yellow were going to appear in the phenotype of the offspring, right? So we were going to see patches of red and yellow. Therefore, we can say that they are coexisting with each other. However, because we're getting a new phenotype, which is orange, we are then going to get incomplete dominance, right? So it is incomplete dominance and not codominance. Okay, 2.4.5. A plant breeder only has plants with orange flowers, right? So here we have red crossed with yellow. It means that the orange flowers are RY, right? Can she produce red offspring from these flowers? Explain your answer. Yes, she can, because if now we breed this orange with another orange like this, we are then going to have RR, RY, RY, and YY. So we can have offspring that are red from the orange flowers. We can also have offspring that are yellow from those orange flowers, right? So it says here, yes, the orange flowers carry one red allele, or they are heterozygous, which is true. If both plants pass on one red allele, the offspring will then be red, which is exactly how it should be. Right, 2.5, it says the diagram shows a cell which has undergone non-disjunction of the chromosomes during meiosis. So as we can see, this would be at the beginning of meiosis, this would be meiosis 1, and then at the end of meiosis 1, and then this would be meiosis 2, and down there we have the end of meiosis 2. Right, now here we started off with 4 chromosomes. Now here we were supposed to have 2 chromosomes each. Right, that did not happen. In this side we have 1, and this side we have 3 instead of 2. So here we have n plus 1, and here we have n minus 1. 1 chromosome short and 1 chromosome extra. Now if we look at A, B, C, D, those are the daughter cells at the end of meiosis. How many chromosomes are they going to have? They're going to have 1, 2, 3, 1, 2, 3, because these arise from this one with an extra chromosome, and here they would then have 1, 1, like that. So an extra one and one that is short. 2.5.1, during which phase of meiosis did this non-disjunction of chromosomes occur? Right, so remember, during anaphase, the chromosomes are supposed to separate. Now, because these chromosomes here, as we can see, they separated here. They failed to separate here. It means that it was still during meiosis 1. Therefore, this is going to be during anaphase 1. Make sure that you do not make that mistake here. Because if you say anaphase 2, you will be marked wrong. Or if you simply say anaphase, you are going to be marked wrong. Make sure that you mention that anaphase 1. Okay, name the type of mutation that will result from non-disjunction of chromosomes. So if chromosomes fail to separate during anaphase, what kind of mutation is that? Because it is changes in the chromosome, we say that it is a chromosomal mutation. 2.5.3, it says, explain the disorder that will result from non-disjunction of chromosome pair 21 in humans. So if that non-disjunction occurs in chromosome pair 21 in humans, what are we going to have? What disorder are we going to have? That is Down syndrome, right? So we will experience Down syndrome. So now it says, explain the disorder that will result in that non-disjunction. So remember, if the chromosomes fail to separate at chromosome pair number 21, it means that when that gamete with an extra chromosome fuses with another gamete that is normal, it is now going to have an extra chromosome. Instead of having 46 chromosomes, it's now going to have 47 chromosomes. Having 47 chromosomes, and if the extra chromosome is on pair 21, will then lead to Down syndrome, which is also known as trisomy 21. Right, so a gamete with an extra copy of chromosome 21 will be formed, the gamete, the sperm cell or the egg cell, the ovum. If this gamete fuses with a normal gamete, it only needs to fuse with a normal gamete to lead to Down syndrome. So mention that. Make sure that you are going to mention that it fuses with a normal gamete, not just any other gamete, but a normal one. A normal gamete will have how many chromosomes? 23 chromosomes. This one with an extra chromosome has 24 chromosomes, right? A zygote with 47 chromosomes or an extra copy of chromosome 21 will be formed. 23 plus 24, it does indeed give us 47 chromosomes. This will lead to Down syndrome. So remember, normal one, 46 chromosomes. Now with an extra chromosome, 47 chromosomes. Remember, you cannot talk about Down syndrome if the question did not mention that this took place on chromosome pair number 21. You can only talk about it if it does say that it's on chromosome 21. Other than that, we cannot talk about it. Okay, 2.5.4, draw a cell to show the chromosome composition after meiosis 2 of the cell division. So now we are going to draw cell A. Let's have a look at cell A. This is cell A, which has 3 chromosomes or unreplicated chromosomes, right? It says here you must draw it to show the chromosome composition after meiosis 2. So we are going to draw a chromosome with 3 unreplicated chromosomes, a cell with 3 unreplicated chromosomes, as you can see here. Now remember, every drawing must have a heading, right? You should get a mark for the heading as well. And then 3 single strands are drawn. It needs to be 3 single strands. It cannot be 3 full chromosomes, but only the single stranded ones, and then the correct size of those 3 chromosomes. It means that we're going to have a look at the sizes that we came up with here. So we can see that we have 2 small and 1 large one. It means that we are going to have 1 large one and 2 small ones, like that, and that is how you are going to get those marks. 2.5.5, it says name the type of cells that will be produced in a male at the end of meiosis. The type of cells that are produced in a male at the end of meiosis are the sperm cells, right? So those are the cells that are produced at the end of meiosis, spermatozoa in males, and it is the ovum in females. Okay, let's have a look at the homework. Question 1.5 of the same paper. It says here, the diagram below shows a short section of a DNA molecule, right? So a DNA molecule is shown there, and then as we can see, we have A, B, and C. Make sure you label these before you can even carry on, right? Now, with this question, as you can see, B, it's a thymine. It means that at A, it's an adenine, right? So you can then say that B is adenine. However, let's assume that this T was not shown, right? And they point at the very same part and they say it's part A and you are expected to name it. You cannot say that B is adenine in that case, right? Don't say that B is adenine. You can just simply say B is a nitrogenous base, because we do not know what type of nitrogenous base it is. However, here, because we have a guide, we can use that T to determine that this nitrogenous base at B is actually an adenine. And then here at A, now we have a sugar, right? So we have a sugar here, and then at C we have those weak hydrogen bonds, right? Now, looking at the questions, the natural shape of DNA, those are some general questions that you can be asked. Make sure that you just go through such information. Identify sugar A, so that would be a deoxyribose sugar. Why is it a deoxyribose sugar? Because we are looking at DNA and it is a deoxyribose nucleic acid, DNA, right? And then the nitrogenous base at B with the full name, and then bond C. Also here, it already says nitrogenous base at B, which is a guide for you, meaning that you cannot say it's a nitrogenous base, but you need to be specific, right? So those are the questions that you will look at and come back with the answers in the next lesson. Now, let's look at our summary. Draw a table if tabulation is required. Guys, make sure that you draw that table. Make sure that you do not lose out on those small marks that are going to just build up and give you a good mark. Pick up on those marks. Even if you cannot give the answers for that question, if you do not know what to differentiate, draw that table. And then if it says differentiate between protein synthesis and transcription of protein synthesis and DNA replication, you're just going to draw your table and then say DNA replication and transcription. That's it. You are going to still get that 1 mark. So make sure that you follow the instructions and avoid losing marks. Right. Remember that a change in the DNA sequence results in a different mRNA and therefore a different amino acids sequence and a different protein. However, you should also remember that as long as the original triplet, codon or anticodon still codes for the same amino acid as this new mutated codon or triplet. It means that the protein formed is still going to remain the same, because if they code for the same amino acid, then there will not be a change in the amino acid sequence, right? So make sure that you do not forget that. Also, another tip that you can look at, when as soon as you come across that question whereby you're given a table, just have a look, go through that table of amino acids and the codons. You will see that sometimes your one amino acid is going to appear more than once. Why? Because it is coded for by different triplets, codons, or anticodons. You will see, for example, alanine, 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 because it's coded for different, by different anticodons. Okay, use XA for the sex-linked characteristics. Also, don't forget XA, X in females, crossed with XA, Y in males. We do not have another X in males, and that Y does not carry any allele, so do not forget that as well. Use the correct format when completing a monohybrid cross. Make sure that your monohybrid cross has everything from P1, phenotypes, genotypes, meiosis, gametes, and then the crossing, as well as the gametes of the genotype of the offspring, which is F1, which should be indicated as well, and then the phenotype of the F1 as well. Now, this question usually has some specific questions to ask, such as the percentage, chance, questions such as the ratio. So you can actually go through that as well. Make sure you supply that so that when you give your answers, you do not lose those marks for not answering the specified questions. You will not lose out on the compulsory marks. Okay, lastly, understand the importance of meiosis in order to understand how non-disjunction leads to Down syndrome. So remember, non-disjunction, chromosomes have failed to separate instead of separating. Now, instead of having two chromosomes, in this example, instead of having two chromosomes, there's now three chromosomes, meaning that the other cell that has lost a chromosome now has one less chromosome. Now, if this cell with three chromosomes, like that, let's say these are the three chromosomes, fuses with another cell with two chromosomes, this is normal, right? Normal. And this one has undergone non-disjunction. If these two come together, what are we going to have? We are going to have 1, 2, 3, 4, 1, 2, 3, 4, 5 instead of 4, right? Remember, this is the n, this is the n plus 1. A normal one and an abnormal one coming together, they're going to give us 2n plus 1, because these are 2n plus the 1. Therefore, that is how we would end up with an individual that has Down syndrome. All right, so in our next lesson, we will be looking at the Eastern Cape Paper 2 question paper for the prelim 2021, and we are going to look at question 3. So here we are going to look at the second part of the paper. We're going to look at the questions that would be making up the very last part of that question paper, which is the third 50-mark part. Okay, we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.